In this video, we will show how to edit a multiple alignment. Since obtaining a multiple alignment is explained in another video, I will create an alignment first without going into detail for that part of the demo. I select all the entries in my database with the Ctrl-A button. Then I create a new comparison. And I display the sequences in the experiments data panel. I calculate a multiple alignment using the default settings. And I zoom out a little to obtain more overview. Now I would like to highlight consensus bases so that it becomes easier to detect gaps and mismatches. To do so, I first create a majority consensus from all sequences. I select the root of the tree. Right-click on the sequence panel and select Create Consensus of Branch. With 50%, I have a majority consensus, which is fine. I right-click again in the sequence panel and select Block Type, Consensus Blocks. For quite similar sequences as in the present example, the automatic multiple alignment works usually fine. But sometimes, particularly at the ends of the sequences, some manual correction might be required. In this region, for instance, I may want to do some manual corrections. I select the region on the sequence that I want to correct. And I drag it until I reach the position that provides the best alignment. Please note that the software contains a multi-step undo and redo function. The result of my action is that there exists a common gap over all sequences in the alignment. I can remove common gaps with Sequence, Edit Alignment, Remove Common Gaps. Also here, I would like to make a change in the alignment. Since the multiple alignment has changed, I will have to recalculate the tree so that it reflects the changes. I press the Cluster Analysis button and select Calculate Cluster Analysis. The tree we wish to calculate is based on our multiple alignment, so I select this option. In the right panel side, there are some additional parameters to calculate the sequence similarity. For example, I can define a specific cost table for base mismatches. I can tell the program to throw away unknown bases when calculating the similarity, and I can specify the penalty for gaps relative to the cost for base mismatches. For phylogenetic purposes, I can apply a distance correction. There are two models available: Jukes and Cantor and Kimura. As an example, I will use the Kimura correction. The option Use Active Zones Only allows me to calculate similarities on selected regions of the multiple alignment. Active zones can only be set if the sequence type has a reference sequence linked to it, which is not the case here. I press Next to move to the next step, which is the cluster analysis step, where I can choose the clustering algorithm. To give the sequence clustering a more phylogenetic meaning, I will use the neighbor joining method, which produces an unrooted tree. As further options in the tree calculation, we have the calculation of standard deviations on the branches and the calculation of branch qualities. If I click on that one, I can see Bootstrap Analysis as one of the options, which I select. Now an additional field appears where I can enter the number of Bootstrap simulations. Usually 100 simulations is fine, so I press Next to start the calculation. In fact, the neighbor joining method produces unrooted trees, but since the comparison window requires the entries to be listed vertically, a routing was done by the software. It is obvious that this routing is not always the one you want to see. Usually, the most distant member or branch of the comparison is chosen to produce the root. If I click on the longest branch in this tree, a red cursor appears, which indicates the place where I want to reroute my tree. To reroute a tree, I press the Reroute button and the selected branch now becomes the root branch. In unrooted trees, branches are sometimes far away from the entry names. A useful feature to link entries to the branches is to display the connection lines. I press the Display Settings button and check the option Show Branch Connection Lines. Please view our other movies to learn about how to create phylogenetic unrooted trees such as maximum parsimony and maximum likelihood trees.